Hello everyone. Insect pest damage is one of the most important factors that affect the crop productivity. The effort of plant breeders to introduce the resistance genes into crop plants from other resistant cultivars and related wild species is a time-consuming and difficult task. However, the technology of genetic engineering has made possible the cloning and introduction of insect pest resistant transgenes into the crop plants. Many insecticidal proteins from diverse sources are available in nature and are effective against agriculturally important pests without causing problems to mammals, beneficial insects, and other organisms. In this module, we will study about the different strategies of insecticide resistance in plants. Let us now look at the outline of the lecture. We will study some of the strategies used for direct gene transfer in plants. These include ballistic transfer, electroporation, microinjection, liposome, PEG or silicon carbide fibers mediated transfer, and the direct DNA uptake during imbibition. Direct gene transfer involves the transformation of plants without involvement of Agrobacterium tumefaciens. There are different strategies used for direct gene transfer. The first one is the ballistic strategy. In this, the gene of interest cloned in a plasmid along with a selectable marker is bombarded on the plant tissue with great force generated by acceleration of helium gas in a vacuum tube. The plasmids are coated onto microcarriers which are gold or tungsten particles of 10 micrometer diameter. The helium gas first strikes a macrocarrier on which the microcarriers are loaded and moves it with high speed, causing it to impact a stop screen. The impact leads to the microcarriers coming off the macrocarrier and bombarding the plant tissue kept at a distance with force. This causes the plasmids to enter cells and nuclei of the plant tissue. The plasmids are integrated into the plant DNA, causing transformation. A variation of this technique is the gene gun, an instrument that does not accelerate helium using vacuum but uses pressure instead. It is portable and can be used to transform intact plants too. The microcarriers are loaded on the inside of the tube through which the pressurized helium passes, and they move with high speed, which causes the plasmids to enter plant cells. Another important strategy is electroporation. A mixture of plant protoplast and plasmid DNA in a suitable medium are placed in cuvettes and exposed to brief high voltage pulses. The DNA passes through the plasma membrane into plant cells. Optimization of transformation procedure is done by varying the voltage pulse intensity, duration of the pulse, concentration of DNA, and the salt concentration in the medium. Typically, a high voltage is applied in the cuvette, because of which the membrane gets charged due to migration of ions from the medium. This causes a change in the orientation of lipids in the membrane, causing pores through which the plasmid can enter. As the voltage decays, the lipids return to their original orientation and the pores close. Electroporation does not harm cells as may happen during ballistic transfer. But regenerating plants from protoplast is often problematic and poses a problem in the use of this technique for some plants that show recalcitrance in regeneration. Microinjection is another direct gene transfer method that was first used to transform animal cells. Here again, plant protoplasts are used for injecting the plasmids directly into the nucleus using a fine glass capillary, while observing the protoplast under a phase contrast microscope. The protoplast is visualized on a screen and the capillary is directed to the nucleus by a micromanipulator that is remotely operated by controlled pressure. Though this technique ensures entry of the plasmid directly into the nucleus, it requires skill and only a few protoplasts can be injected manually. However, subsequent handling of the protoplast in both electroporation or microinjection is similar and involves plating the protoplast on a regenerating medium and then subjecting them to selection by adding an appropriate antibiotic depending on the selectable marker used. Regenerating calli are then allowed to grow into plants. 3 more strategies have been used for direct gene transfer by various laboratories. All of them use protoplast for transformation, which is a problem because regeneration from protoplast is not easy. Silicon fiber whiskers puncture the membrane through which the DNA can enter. The whiskers are mixed with plasmid DNA and function as numerous needles facilitating DNA delivery by cell perforation and abrasion. In the PEG method, use of high PEG concentration in the presence of calcium ions causes a change in membrane permeability due to alterations in the properties of the lipid bilayer, hence allowing the DNA to enter. Liposomes are artificially made lipid vesicles that spontaneously interact with the plasma membrane of protoplasts. Effective delivery of their DNA content within the protoplast is induced by treatments with PEG or DMSO. On comparison of these methods with agrobacterium mediated transformation, it is seen that the latter are more expensive and can cause multiple insertions of the gene of interest. On the other hand, the agrobacterium DNA delivery method ensures single copy insertion, but the process is slower. The module can be summarized as: many insecticidal proteins are available in nature and are effective against agriculturally important pests. They include crystal proteins and vegetative induced proteins produced by Bacillus thuringiensis. In addition, plants produce proteinase inhibitors, alpha amylase inhibitors, lectins and chitinases as a part of the defense arsenal. Production of transgenic plants expressing genes coding for these insecticidal proteins has led to production of expression of insect resistance in the transgenic plants and offers a way of protecting plants against insect infestation without the use of pesticides. As a result, transgenic crops producing Bt toxin have been widely commercialized. However, there are several risks in the cultivation of such transgenic plants since they may pose ecological and health hazards. Thank you.